As Sandra introduced myself, my name is Stephanie. I'm a data scientist here at Biosymmetrics, specializing in computer vision. The talk for today will be a machine learning-driven phenotyping platform for in vivo target validation and precision medicine development. So slightly different from all the talks that you've received today. So our mission at Biosymmetrics is to translate data into discoveries. Biosymmetrics is a phenomics-driven drug discovery company that generates data-driven insights from experimental and biomedical data using machine learning to translate complex human disease biology to advance precision medicines. So overall, I'll be talking more about the AI computer vision aspects of our platform. Okay, so a quick overview of what we'll be going over today, an introduction into our model organism. So we use live zebrafish, the challenges associated with using zebrafish models, in terms of live data, how we adapt to that, the inconsistencies with live data, the overall methodology of our pipeline, and then going into our results and conclusions, and then future directions. So zebrafish, most of you might not have heard about zebrafish before, but it's our first model organism, and the image right there is an example of a fully grown zebrafish. It is transparent in this picture specifically. It doesn't look very transparent, but nonetheless, when it is growing from 2 days, 3 days, whatever it might be, it's fully transparent, and this allows us to take a look at it as it's growing, developing its heart, brains, etc., making it a great model for development in drug discovery. So the zebrafish itself is very fast in terms of target validation. From 48 hours from the initial CRISPR injection, you're able to see some sort of phenotype. So what a CRISPR injection is, it's just a tool that basically disrupts a certain gene, and that gene will eventually show a phenotype of interest. So whether it be heart abnormalities or brain dysfunction, whatever it might be, that's what that specific tool does. The overall zebrafish, let's say, apparatus is fairly scalable. So you can basically screen about up to 300 fish in a single 96-well plate, which makes it a great model for high throughput and reproducibility purposes. So in terms of why can we look at zebrafish to humans, surprisingly, humans and zebrafish are very similar. About 70% of there is about 70% human genetic conservation. About 80% of those genes are associated with human disease. So in the literature as well, you can also see that there are a lot of translatability between cardio and neuro programs within zebrafish and humans, whereas the mouse model has indeed failed. So all of this in combination makes zebrafish a great model organism for us to look at, specifically for cardiovascular purposes. Okay, a little bit too far. Okay, so the overall objective was to basically use machine learning and computer vision tools to accelerate phenotypic analysis of zebrafish imaging data. So that image up there on the screen is a single well. So again, there's 96 of these in a single plate, but this image itself is a single well of a zebrafish. This is a disoriented well. There's no consistency in the data. You can see that certain fish look different from the others. Some have the embryonic sac, some do not. And you can also see within that quick 3-second video, there was quite slight movement within the zebrafish. So you can imagine that over a period of 3 to 4 minutes, there would be a lot of sporadic movements, making the actual image analysis portion of the analysis quite difficult. So I'll just go over a bit of terminology for the folks that don't necessarily have a biological background. So what is a phenotype? A phenotype is basically an observable trait. Anything that you can see with your eyes, whether it be hair color, height, disease, whatever it might be, that is what we generally describe as a phenotype. So in those two videos there, you can see a control zebrafish as well as a mutant. A control zebrafish is basically a healthy living zebrafish with no physical observable traits of any abnormalities. A mutant zebrafish is what you see also there. There's an enlarged heart region and abnormal blood circulation. And this is basically done using that tool I spoke about earlier, which was the CRISPR tool. For this specific video, we've disrupted the gene called MYH7, and that caused this specific phenotype, which is fairly important when we want to quantify said cardiac phenotypes. So just taking a step forward into the actual abstract or case study, now that we kind of are hopefully on the same page in terms of the biology lingo. So the overall study was basically to develop a zebrafish screening platform that has three specific features. One, a custom 3D printed mounting platform, and I'll go into that. This is supposed to help us with our data inconsistencies. Two, a fine-tuned deep learning instance segmentation model, and I'll explain again why we needed a segmentation model. And then three, a computer vision post-analysis pipeline for quantitative analysis. So these three components all together is what our zebrafish screening platform is built upon. And for this specific talk, we'll only be looking at the cardiac phenotypes. So working with zebrafish, now that we know what a zebrafish is, for most of you, you saw a video where the zebrafish was moving. There is a good amount of inconsistency in the data, but before going into the actual data, looking at the cardiac phenotypes, humans and zebrafish have very similar ECG patterns. And what an ECG is, it's basically a way to measure your heart rate. It's an electrocardiogram. Usually humans, you hook yourself up to one, and you can get some sort of signal out of it. Unfortunately, we cannot do this with zebrafish. So the way we actually compute the heart rate is by looking at the video itself. So the video there is just a high-resolution video of a healthy zebrafish, and you can see that there's some pixel intensity changes in the heart area, and that is basically what we quantify. So we look at the pixel intensity in combination with Fourier transform to produce an ECG. And again, I'll go into that in more detail later on. Okay, so the challenges with this data set are as follows. So if you're not aware, medical data sets come with its own gigantic amount of challenges. One being image parameters, vendors. Every lab has a different vendor, every lab has a different protocol, and this creates a gigantic mess in terms of data. But with live organisms, this adds another layer of complexity. You're not able to orient the fish. The fish might move in different directions. They might even overlap, making that transparentness of that fish unusable. You can't necessarily use that data if there's two fish on top of one another. So again, any type of response, so if the animal is injected with some sort of injection, or if there's a chemical compound really inside that well, the animal might move in response to that, causing orientation issues. And there's also a large data imbalance. So typically obtaining healthy zebrafish is fairly easy, but when it comes to abnormal disease characteristics, which our model should see, that can become challenging, and that basically gives us a low yield of data, and some of these treatments can become toxic, so it gives us a dead zebrafish, which isn't necessarily usable. So the overall objective of the first step was to develop an orientation strategy so that we can capture animals that have a consistent orientations and do not move for the most part, so no sporadic movements. And then the number of animals within a single well should be maximized. So here we have 3 different images, all from different laboratories, or sorry, the top 2 are from one laboratory and the bottom are from another. And you can see the differences in the data just from this image here. So some of them have fish that are going towards the outer well, it becomes darker. Some are still in the embryonic sac. In the bottom fish, you can see that there's a bit of overlapping occurring, and you can also see the different phenotypes within the bottom one as well. So the overall methodology is what we're going to be speaking about next, and this includes the 3 components that I spoke about earlier. So first, our objective is to quantify a cardiac metric to aid in the classification of cardiac phenotypes. So first we have our custom stamps at the very top. That helps us orient the fish. That data is fed into our model is trained on, and then eventually, when we do want results, we're able to pull out some images of segmented organs, and then using that, we can quantify a vast amount of phenotypes. So things like heart rate, morphology, blood flow, ventricular volume are all examples of phenotypes that we can report out. So starting with the 3D stamps, we designed 3D stamps to standardize the zebrafish orientation. So the top image there is a 96 well plate stamp, and what this basically is it's exactly what you might think a 3-year-old plays with, a stamp. So you take this stamp, you place it into the well, or the plate, sorry, and that will imprint the agros inside of the plates to have that little mount for the zebrafish to be placed upon. So once that's done, the technician would be able to place the zebrafish onto these different mounts, and then the zebrafish should not move too much. You can still see blood flow, heart rate, etc., but the sporadic movements are basically gone. So the benefits of these, it's consistent in orientation, it does not move, and now we're able to maximize the number of fish without having to throw away any valuable data. So we have 2 stamps. One is a novel stamp that our company had developed, which is a ventral stamp, and this is where the fish are facing upwards. So you're placing the fish up towards the microscope, and you're able to see the ventricular area in general, and this would help with locating the ventricle, pericardium, things of that nature. Now, if we're looking at our second stamp, it's a lateral stamp, the bottom image over there. And this is basically great for blood flow. So if you'd like to see the overall circulation within the zebrafish, you're able to do so with that specific stamp. Oh no. Okay. So once we've standardized our imaging setup, after imaging acquisition, we basically needed a way to label all these different organs within the animal. And doing this by hand is very time consuming. And again, there's about 9 organs that we segment. That would take forever, and that's where we came upon thinking about leveraging pre-trained models to accelerate organ segmentation. So we have a couple issues with training from scratch. We don't have enough data, and that's why we looked to transfer learning. So we used Detectron-2, which is an object detection and segmentation library by Facebook AI Research. It provides detection and segmentation algorithms, and they're all pre-trained using the COCO dataset and ImageNet. So the pre-trained model that we selected after a little bit of experimentation and investigation was the MaskRCNN ResNet 101 with the FPN backbone. And as you can see in the image there, we trained on about 2,400 images, and each image itself had about 7 to 9 organ labels or regions of interest, and then that was used to train the model. Oh. Okay. So the overall objective of our instant segmentation model was to segment regions of the model organism which can be later used to quantify phenotypic parameters. So cardiac organ segmentation was based, for this specific model, we were looking at the cardiac phenotypes, and for that there was about 7 to 9 organs that we were interested in between the two different orientation. So in ventral, you had the ventricle, zebrafish. Lateral, you had notochord, myotome, finfold, zebrafish. And then there was a bunch of labels that were also common labels like eye, head, yolk sac, pericardium. And all of these organs were later used to calculate or quantify different cardiac parameters, all listed below. So the overall model, on the test set, this figure here is how it performed. So you have our score of average precision, and then all the 9 organs labeled at the bottom. And you can see that for the most part, the model did fairly well. Most of the labels achieved a score of above 80%. However, there is some improvement. Things like the myotome, the finfold, were scored lower, and this is something that we do need to improve upon. But for the aspects of this project, we were more invested in the yolk sac, the zebrafish, pericardium, those regions, and that's why we continued onwards. So the overall goal, again, is to leverage existing segmentation models with computer vision and image analysis in post-processing to basically accelerate our overall drug discovery process. Doing this without AI might take months because of the manual analysis that it's required to look at each zebrafish to see what types of phenotypes is visibly there. So in post-processing, I'm going to go through a few examples of the cardiac parameters that we looked at. And using the organ segmentation results, we looked at heart rate. So in terms of heart rate, looking at the pericardium region or the yolk sac region, or that's basically the body of the zebrafish in the ventral format. Looking at that in combination with pixel intensity changes and Fourier transform, we're able to create a pseudo ECG, if you will. And this will quantify our heart rate. So here you have an image of, or a video of a healthy zebrafish at the very top. And at the bottom, you have a zebrafish with an abnormality, a trembler type of heart rate. Next is blood flow. Blood flow is extremely important in terms of cardiac phenotypes. This looks at the overall flow and circulation of the zebrafish in general. And what we do here is look, there's 2 different stamps that we can look at, lateral and ventral. Ventral is the one at the top, where you take the entire zebrafish, you look at the overall flow and pixel intensity changes throughout the zebrafish itself. And then the bottom is a lateral view. So this is where you can take the zebrafish. In this specific example, it was a zebrafish mask. You can also take the notochord, myotome, whatever you're interested in, and then look at circulation that way as well. And this specific video looks at using optical flow to false colorize the flow in general. Finally, we have ventricular volume. Ventricular volume and edema-related phenotypes. Okay, so looking at edema-related phenotypes, what edema basically means is basically it's some sort of blood pooling or blood circulation issue. So typically edema would look kind of like a bloated body, like you see at the top few zebrafish there. And that's where we take a look at the organ segmentation results, look at the overall zebrafish or the body or the yolk sac, and we can point out these morphological changes. There is some blood pooling in the last zebrafish. You can kind of see a little bit of red on the end over there, and that again is reported out as a metric. Finally, we have ventricular volume. And what ventricular volume is it's the amount of blood flowing through a ventricle or the heart chamber. And this is quantified using ejection fraction, stroke volume, cardiac output, things of that nature. And we use the ventricle segmentation to report that out throughout all the frames of the video. So looking into the results of these cardiac phenotypes, we're able to basically look at ground truths. So having an expert manually quantify heart rate, then comparing that. We're also able to use cardiac compounds where we can mimic a expected result and see if our method is following just that. And then we're also able to use that CRISPR tool to see if we can also get an expected output. So the first one here is heart rate, where we're looking at the heart rate of each embryo, and an expert would look at the videos. They would annotate, or not annotate, but quantify the heart rate, and then we would use our automatic approach to see how correlated it is. And for this specific case, we looked at controls, so healthy zebrafish, as well as induced with nifedipine, and nifedipine basically lowers the heart rate. And as you can see in the image there, we got a correlation of 0.998, so high correlation, meaning that our heart rate quantification was fairly accurate. Now looking at CRISPR loss of function. So again, CRISPRs are basically a tool to disrupt a certain gene. For this specific gene, we looked at MYH7, LMNA, and TNNT2A. All of them have a decreased output in heart rate and ejection fraction. Ejection fraction is basically how well the heart is pumping out blood. So if all of that is supposed to be expected to be in the decreasing direction, and that's exactly what our automated approach reports out. Now, we also looked at the cardiac compounds. So in this specific case, we looked at nifedipine, and again, it's supposed to have a decreasing output in terms of ejection fraction, heart rate, and cardiac output, and that's exactly what you can see here. And of course, all of these results were analyzed by experts to make sure that we are reporting out valid results with accuracy as well. So in terms of this, nifedipine is a chemical channel blocker clinically used for treatment of angina and high blood pressure. So again, the expected results are as follows. Now, we didn't stop just there. We also, this would say, we also looked at dose responses. So we wanted to ensure that whatever chemical compounds we're looking at, as we increase the dose, do we see a similar trend? So in terms of this image here, you can see that as the dose is increased, the trend still follows. And because we did a decreasing cardiac output or heart rate, we also wanted to see if the increasing is also a trend that follows. So here we see that there's an increasing dose, increasing concentration. We're able to see that the expected output was increased heart rate, cardiac output, and ejection fraction, and that is exactly what happens here. And there's a nice video for you. And to summarize, data variability is a fairly large problem in the medical field in general, but when it comes to live organisms, it is fairly difficult to deal with. So there has to be imaging protocols to standardize this, and we basically used stamps to do such. And we're also leveraging segmentation models to accelerate the overall process of quantifying these zebrafish phenotypes. Now, in our future directions, we are planning on looking at epilepsy models. This basically looks at stimuli and whether or not we're able to quantify a behavior or a seizure-like movement within a specific epilepsy model. It's just a fun little video for you. And I wanted to say a big thank you to our team. None of this would have been possible without them. With that being said, I could take some questions now.
Thank you very much, Steph, for your presentation today. Now we are open for some questions. There are some people with mics around the room. Yeah.
Thank you. Kian from Recursion Pharmaceuticals. We're quite interested in phenomics as well. I had a question about your low data regime. So you said, if I recall correctly, you had about 2,400 labeled examples for your segmentation. Mm-hmm. I imagine you might have more images which are unlabeled. So I was wondering if you've considered doing sort of self-supervised learning techniques on those images. And then as a follow-up to that, it seems like your downstream tasks, are fairly sort of far removed from the deep learning training objectives. So I wonder if you have any insights about the challenges with benchmarking those deep learning models that happen so upstream versus, these final downstream metrics. Thank you.
Great question, thank you. So I'll try to address the first one. I think the first one was revolving, sorry, what was the first one? Yes, so that is something that's a reiterative approach that we're working on, looking at using the incoming data stream to basically train our model, get outputs, and then use that outputs, fine tweak them, continue on the training process, and we're also looking into semi-supervised learning as well. In terms of the second question, which, if I'm not mistaken, no, I am mistaken. What was the second question?
Benchmarking these deep learning models is where the downstream tasks are far removed from perhaps the loss.
Yes, so I guess you could say the more complex deep learning models, they require a vast amount of data, and that is where we struggle with. We don't have tens and thousands of data to kind of feed our model, and if we did, from pulling out frames and videos, that would cause a bias, and that's where we're kind of struggling a little bit, and that is a major reason why we're looking more to transfer learning and pre-trained models rather than building one from scratch per se.